starting with Metaphase 1. Now the diagrammatic view of Metaphase 1 will be somewhat like this, where we are going to draw the spindle apparatus first. Astral rays, spindle fibers, and obviously the chromosomes at the equatorial plate. Now here what we have to keep in mind is the way we have shown crossing over and chiasmata formation, and then the counterparts exchanged, we are going to show them here. Okay, and same goes for these. Their tips were exchanged, remember? So this is chromosomes in two equatorial plates. Equatorial plate, or we call it metaphasic plate. So this is the appearance of chromosomes. Rest we all know. Spindle fibers are there, centrioles are paired up, astral ray formation takes place, nucleolar material, that is the nuclear membrane disintegrates, right? So here we are writing the key events. Number 1, the bivalence or tetrads are arranged at the equator. At the equator of spindle. Alright. And number 2, the centromeres are aligned in 2 rows. Clear? Moving to the second one, Anaphase stage. In this Anaphase stage, the chromosomes will start receding towards the ends. Again, we are making the same spindle apparatus. Every time we have to make it without forgetting. And here we show that how the chromosomes will recede towards the opposite poles. Now, this one like this. Remember, we discussed that these move in I, J, L and V shapes. So 2 of them have got the exchanged part, and next 2 and the next 2 are simple. See, this one here, this one here. As we have shown little differentiation, that one is receding and the other one is little away. So in this anaphase, we write that members of every homologous pair of chromosomes these are homologous pairs, right? Members of every homologous pair of chromosomes separate from each other and start moving to opposite poles. Number 2. The important key feature here is the number of chromosomes become half the number of chromosomes of parent cells. The number of chromosomes become half the number of chromosomes of parent cell. Clear? Here occurs the reductional division. Then we say telophase 1. In telophase 1, we see that chromosomes undergo dissolution and they have moved to the opposite poles where the nuclear membrane is regenerating and the chromosome chromatin thread will start appearing. These chromosomes one pair with each. Now this is going to dissolute the spindle fiber, spindle apparatus. Alright, here we say chromosomes have reached the poles. And secondly, nuclear membrane forms around the chromosomes, clusters at each pole. Okay, so this is what is observed. What is expected after that? Cytokinesis. When the chromosomes have reached at the poles and nuclear membrane has started appearing, or then slowly it completes, then what is expected? The first nuclear division has taken place. Now the two daughter cells are formed, right? This cytokinesis generally follows. It follows after the first nuclear division. Then only the two cells will start dividing further, right? Then comes a stage which is known as interkinesis. Interval between the nuclear stage. Kinesis is nuclear division, inter means the interval. It is similar to a short interphase. A short interphase between Meiosis 1 and Meiosis 2. So what happens here? Here keep in mind, no replication of DNA, because we are dividing the material and this is only getting copied. So no replication of DNA, and sometimes if not important, this interface can be missed out. It may be absent. So not necessary that it has to be present. It may be absent, and then begins with the Meiosis 2. Then Meiosis 2 starts from here. Alright, now Meiosis 2, the key events will remain the same. It is similar to mitosis, okay, and is known as equational division, as I told you in the beginning, because here the number of chromosomes gets equated. So we start with again karyokinesis, and karyokinesis means, by now you know karyokinesis stages. Meiosis 2, that is prophase 2, metaphase, Anaphase and Telophase. Right, karyokinesis. Let's begin with karyokinesis stages. When we say prophase 2, here the prophase 2 is simpler structure, again showing the nuclear membrane disintegrating, showing the spindle apparatus. Its formation is being done, astral ray formation. Please use different color for everything. And then the chromosomes. This one and the other one. This one and this one. This much we have learnt. How have they become colorful? So we are going to begin with this prophase 2. So what are the key events? The sister chromatids of each chromosome start condensing and become shorter. Means sister chromatids condense, become shorter and aligned. And aligned in pairs. Okay? Then moving to Metaphase 2. After prophase, always the metaphase. Spindle fiber is complete. Fibers are these, and apparatus is when we say this entire structure containing centrioles and astral rays, fibers. We draw the chromosomes here, where they are aligned at the equator. Okay, here are the ones. Right, they are at the equatorial plate. So, metaphase 2 at the equatorial plate. What do we write for this? We write that a bipolar spindle is completely formed. A bipolar spindle is completely formed at both the ends, right? Opposite ends. Then chromosomes become arranged at the equator of the spindle. Clear? It's quite visible here. Then moving to Anaphase 2, the third stage. Same way, Anaphase 2 will show the receding of the chromosomes towards the opposite poles. And the chromosomes will move away with the same I, J, V shapes. Okay? Any shape, I, J, L, V, you can name. Now this is L kind of shape, V kind of shape. The centromere of each chromosome divides into two. What does it mean? It means this. We had this now, and this is the centromere. we had this structure initially here, so it is going to divide into 2. The centromere is going to divide into 2. Okay, and the sister chromatids separate and move to the opposite poles. Sister chromatids separate and move to the opposite poles, following the same shapes, right? And how do they move to the opposite poles? As daughter chromosomes. How? As daughter chromosomes. Alright, coming to the end, that is the telophase stage, the 4th stage. In telophase 2, the structure will appear somewhat like this. Here we see reappearance of nuclear membrane, nucleolus, and the chromosomes at the opposite poles. Right? So this is the structure. We'll show it like this, somewhat. Here again the nuclear material, that is the nucleolus, nuclear membrane reappears, but when we talk about the spindle apparatus, it is again fading. Astral ray spindle apparatus, it is fading because cell has come now to division, cytokinesis. In animal cell, it is the invagination of the furrow. Furrow gets deepen, 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 and further it divides, right? Remember? And ultimately, the telophase events are chromosomes have reached the poles. Chromosomes have reached the poles. What will they do? Start uncoiling. Remember? Coiling and uncoiling. Uncoiling के बाद क्या बनते हैं? Becomes thin. Do mention the nuclear part also. Nuclear membrane, nucleolus, it starts reappearing. To form daughter nuclei. Okay, and after that what occurs? Cytokinesis. Cytokinesis means now the nuclei are separated into daughter cells. Daughter cells, and each daughter cell or dyad, it forms 2 daughter cells further. Okay, hence at the end of meiosis we get a diploid cell forming 4 haploid daughter nuclei. Okay, forming 4 daughter nuclei. How come? 1 from each daughter cell. 2 daughter cells are formed and 1 from each daughter cell. Ultimately at the end we have got 4 daughter nuclei. Once we have reached here, there must be a significance. The significance of meiosis. What is that? Number 1, ensures the maintenance of constant chromosome number. It is very, very important that it maintains the constant chromosome number. Characteristic of a particular species, generation after generation. The number remains chromosome number remains constant. Number 2. Whenever the crossing over takes place, this crossing over that has taken place, it introduces variations. Right? Variations in offsprings, and variations are always the raw material of evolution. Clear? So variations automatically lead to evolution. So whenever meiosis is taking place, it is basically in meiosis that we have learned that chromosome number remains constant and the type of chromosomes in a species that remains as it is. The number remains as it is, then maintenance of this number by halving the same remains as it is. Then assortment of chromosomes. It causes reshuffling of chromosomes and the traits are controlled by them. Gamete formation is essential, which is for sexual reproduction. So this is the only event which helps in gametes formation with haploid number. No doubt gamete formation is taking place in other as well, but here that is essential for reproduction. Sexual reproduction with half the number is in meiosis. Alright, this is all about the chapter.